Hey guys, just a little talk about viruses today. Shouldn't take too very long. It does include a special edition for the coronavirus. Just some pictures seen from the 1918 flu pandemic. An especially nasty bug, worse than what we got now, definitely. A lot more deadly. And deadly to people that weren't old. Seen from the 2003 SARS scare. We also had a virus back then. You guys were too young to even know anything about it, is my guess. This is maybe not entirely representative of the COVID-19 scare, but because there's nobody playing Animal Crossing as far as I can tell. When we look at viruses, we want to start at the very beginning. We want to start thinking about how big they are. They are very small, right? They're just molecular complexes that F with your S. That's what they are. They're a simple combination of molecules. It's a handful of proteins and some nucleic acids. Occasionally a little bit of a phospholipid bilayer, but that's about it. Not anything you would call a cell or anything, but very small. The scale I usually come up with is, the classroom is, if the classroom is the size of a eukaryotic cell, then a bacterial cell would be like a soccer ball or a football. And then a virus would be like, a Tic Tac or like an ibuprofen tablet. So they're much, much smaller than cells. This is just a cool electron micrograph of a bacteriophage attacking an E. coli cell. A phage is just a virus that infects bacteria. We call them phages. Sometimes with plants, you see these kind of spotty defenses. A part of this is infection by the virus, but also we've talked about the ways that plants defend themselves. Sometimes they will let the cells die. But there are a lot of plants that we cultivate that we deliberately have infected these strains with viruses because they make interesting color patterns. This is not a positive thing. If your tomatoes look like that, that's problematic. But some lovely tulips. The logic of viruses is a little bit like a lot of simple organisms. We don't consider viruses organisms, but they do behave in a very similar fashion. Huge numbers of offspring. And not careful with the proofreading of their DNA, not careful with maintaining the integrity of their code. So they get a lot of variation this way. So they mutate very, very easily. They never proofread. If they make a mistake, they just let it go. This means that they have a huge amount of variation. And that means they have the fuel for evolution. So you shouldn't think, why would a virus do something like that, right? They're not planning. What they're doing is trying everything. And almost nothing is working as far as the variations go. Most of the variations end up being non-viable viruses. But sometimes you get a virus that does something entirely new or is able to infect an entirely new cell or is maybe more harmful or maybe more easily passed, survives longer in the air, survives longer on surfaces, things like that. They're just going to stumble across these mutations. And as they stumble across them, the ones that are most successful, those are the ones we get to see. So what we are always seeing with viruses is the latest model of any particular family of virus, the most effective one now. And keep in mind that not only are they evolving, but we are too. And we're evolving our defenses. And all living things on Earth are evolving defenses against these viruses. So it's a constant arms race. So when we look at a virus, we've got a basic idea. We've got a protein coat and we've got a nucleic acid on the inside. And they come in lots of different shapes. Sometimes they're cylindrical, sometimes it's an icosahedron, right? The 20-sided. They have these interesting kind of spidery looking ones that are very kind of creepy. I once had a student tell me that he had a nightmare that a bunch of these were chasing them and they all had my head on them. Scary nightmare. And you have these ones that take a little piece of the phospholipid bilayer from the cell that they've infected. And so you end up with this little piece of membrane around them. They still are surrounded by a protein coat, and the nucleic acid on the inside is also surrounded by a protein coat. And this is our coronavirus is like this. So is HIV. It's also one of these that takes a little bit of the membrane. So there's actually a wide range of viruses that all follow the same basic format, right? It's a nucleic acid with a protein around the outside of it. And the nucleic acid can be double-stranded DNA, it can be single-stranded DNA, it can be double-stranded RNA, it can be single-stranded RNA that serves as mRNA, and that's sometimes called a plus RNA virus. And then there are single-stranded RNAs that are templates for making the messenger RNA or the transcripts, and that's a minus RNA. Basically, it's antisense. And then you also have single-stranded RNAs that are used to make DNA, which is a little bit interesting, but that's called a retrovirus. And so retro because they do part of the central dogma in reverse. Instead of going from DNA to RNA, they take RNA and use it to make DNA. And the enzyme that it uses to do that is called reverse transcriptase. It does transcription in reverse. All of these, what they have in common, it's a nucleic acid surrounded by a protein coat. That's what viruses are. Big molecular complexes that are always in this kind of same basic format. A nucleic acid with a protein coat, and they get into cells, hijack the machinery, and really F with your S. So we'll look at a really simplified kind of model of how a virus works. So here's our virus. The purple is the protein coat. The light blue is this nucleic acid inside. It looks like DNA because it's double-stranded, but it doesn't really matter. It's always going to work like this. It's going to get into the cell in some way. It does this by binding a receptor on the cell, which is why they're not able to jump from one species to another, or why they often attack only a specific type of tissue in an organism. It's because they are binding a specific protein receptor. And their nucleic acid is used to make copies of itself through some process. And it's also used to make protein. So we're doing two different things, making copies of the nucleic acid and making copies of the protein coat. And then we're going to assemble those together and then somehow have those protein-coated nucleic acids, those new viruses, leave the cell. Sometimes they rupture the cell and spill out and go everywhere and the cell is dead. Other times the cell stays intact. It'll take out all the rest of the function of that cell and turn it into a virus factory and just continually crank out these viruses. To keep it simple, we'll look at bacteria, but keep in mind that there are eukaryotic versions of what I'm showing you in these next two models here. So the lytic stage of a bacteriophage, lit, right, is the same word root as lyse, L-Y-S. It just means to break or to cut or to rupture. And so what these guys are going to do, they attach to the outside of the cell, inject their nucleic acid, use that nucleic acid, like I said, to make copies of the nucleic acid and to make a bunch of the protein coats. And then they will assemble and then rupture the cell. And now each of these can go and infect another cell just like this. So this is the lytic cycle where they infect a cell, make copies of themselves, and then bust the cell open and go do the same thing to other cells. So this diagram shows both the lytic and the lysogenic. The lytic cycle is over here on the left-hand side. But the lysogenic, what it does is injects its nucleic acid, and the nucleic acid gets integrated into the host chromosome, either as a copy of the nucleic acid or as the actual piece of DNA. If it is a DNA virus, sometimes that can happen. This will be copied when the cell goes through binary fission, or in the case of eukaryotes, mitosis. It'll get copied, it'll get transmitted to the daughter cells. They will all have that. And then some signal occurs that tells this virus, hey, it's time to be active. And it will switch over and do the lytic cycle again. So lysogenic, it just basically is sleeping in the genome, just hanging out there waiting for the right conditions. And there's a lot of viruses that do this both in prokaryotes and eukaryotes. What's interesting is that sometimes as the virus is sitting in the cell, a mutation occurs and you end up with a non-viable virus still in the genome. Our genome is full of these things, of old viruses. Some of the virus, we have clear evidence that this virus infected an early primate and all the primates have this old virus sitting in there. I've kind of spoiled this, but many viruses have what's called a viral envelope. And this is just a little bit of cell membrane that surrounds the virus. And the way this is formed is a lot like the way we butt a vesicle off of the ER. Remember we have the ER and we butt a vesicle off. Well, in this case, instead of butting off of the ER, we're going from cytoplasm to the outside. So we're actually butting off the cell membrane and making what is called an exovesicle. And so this is a lot like a vesicle inside your cell, but it's outside of the cell. And it's what the virus uses to get around. Our coronavirus this year, the COVID-19, this has this same envelope. HIV also has this envelope. It's very similar. But it's got these spike proteins around the outside. It's got protein coat on its nucleic acids. And so here's the nucleic acid with the protein coat around it. Here's the envelope. and here are the spike proteins on the outside, and those are the things that are going to bind the receptors of the cell that it's going to infect. This is one of my favorite high fives ever. Those guys are not cowboys. I know cowboys. They're not cowboys. So here's an RNA virus with its envelope. So what's going to happen is these guys are going to bind a receptor on the outside of this cell, and that's what's going to tell it, oh, hey, we're going to be incorporated here. And we'll just fuse this membrane to this like we're fusing a vesicle. And so the membrane becomes continuous with this. You end up with these proteins in the membrane. We're also going to produce more using the ER and the Golgi. So that we can package more viruses like this. But it's an RNA one. And so in this case, this is, it looks like it is an RNA minus because this is antisense. We're using it to make the transcripts, the mRNA. We use that to make all the proteins that the virus needs to make its protein coat and to make these membrane proteins. We're making copies of the genome, in this case, the RNA genome. We're going to package that into these guys and then just butt off these vesicles, these exovesicles filled with the virus so that the cell can remain intact. It's no longer doing whatever its normal function is. Now it just does this. It just builds these viruses and keeps butting them off. So coronaviruses, COVID-19 just means, it's just short for coronavirus disease. And of course, it first appeared in 2019. But it's a coronavirus, meaning it has this crown, this halo of these spike proteins around the outside that are visible with a electron microscope. It's a plus RNA protein, which means it uses its RNA as a transcript, and we'll look at that here in a minute. And it has these spike proteins that bind the receptors in the host cell, and it has a bit of cell membrane. So this is, like I said, this is basically a phospholipid bilayer. So this diagram came from a paper describing coronaviruses back after the SARS outbreak. SARS is also a coronavirus. So the virus has the spike proteins. You can see here, it's got spike proteins that are binding receptors on the cell. That causes this to fuse. So we end up with our nucleic acid inside of the cell. Ribosomes are going to do translation on a piece of that transcript, on the piece of that RNA, and make this enzyme called replicase. Now, the replicase is an enzyme that takes RNA and makes an RNA copy of it. This is not something your cells normally do. Normally, if we're going to make RNA, we make it from a copy of DNA. So it has to make this enzyme itself. It uses the cell's ribosomes to make this replicase, and that allows it to make a RNA minus, which can then be used for transcription. So it works like the DNA. This RNA minus is now going to work like the DNA so that we can make all of the little RNAs. These are all messenger RNAs, are all transcripts for all of the proteins that are necessary for this virus. And all of those came from this RNA minus that we made from the RNA plus using that replicase. We're also going to make copies of this using that. We're going to use that replicase again. We're going to turn this RNA minus into RNA plus, and that's the RNA we're going to put into our capsule again so that we can secrete this virus again and have it go and infect other cells. Some interesting things happen here. So it builds a lot of these proteins. Of course, these spike proteins all have to be built through a membrane. They are built into the ER. And so it's going to build these membrane proteins into the ER. It's going to make a vesicle into the inside of the ER by surrounding that RNA copy that we made. And then so when it buds off of the ER, it's going to be in a smooth vesicle without the spikes. But then this smooth vesicle is going to fuse with the outside membrane of the cell and dump the full completed virus with its envelope out of the cell. So it's a little bit different because it actually doesn't, grab this section from the membrane of the cell. It grabs it from the membrane of the ER, which is interesting. So the RNA minus is used as a template strand to make transcripts. All right, the RNA plus is sense. It has codons. The RNA minus has it's like I said, it's like the template strand of the DNA. A lot of these proteins have to be made into the membrane of the ER because they are going to be part of that envelope. When we look at the spike protein, the S1 bit on the very end of it, that's the part that binds the receptors of the cells it's going to infect. And this place mutates more often than every place else in the genome of the of the virus. Everything else of the virus has a mutation rate that's pretty high, but that bit really mutates a lot. And this is really helpful to the virus because it means it's always trying new things for getting into cells. It's like, there are keys that open locks, and it is trying new keys. It's making new patterns of keys and trying them because you never know when something's going to work to get into a new cell that you couldn't get into before. And this is how you jump species. This is how you affect different tissues. This is how you spread. It makes it so this virus can move from one species to another. So another pretty nasty virus is HIV, human immunodeficiency virus, which of course causes AIDS. And it also has the envelope the way that the coronavirus does. It has the protein coat, it has a nucleic acid, in this case it's RNA, and it has this little enzyme here that comes with it. There's a protein that comes with it. Before it can transcribe or translate anything, it has a protein that was packaged with it to begin with. And this protein is a reverse transcriptase that I was talking about before. So what it can do is it can take this RNA code and then reverse transcript it into DNA. So we're going to use this to make DNA. And then that DNA can be incorporated into the host cell genome. And so you can see here's the cell is, fusing its membrane, just like the other virus did. Now we've got this RNA, we've got this reverse transcriptase. So we use the reverse transcriptase to make a single strand of DNA. We synthesize a second strand of DNA off of that, and then that gets spliced into the host cell's genome. It just gets stuck into a chromosome somewhere. And so from there, it can make transcripts. Those transcripts include codes for proteins of the virus. Everything that the virus has, those transcripts are coming from the host chromosome from this copy of the virus in there. And so we can have mRNA, we can have the RNA copy of the virus code. We get the protein coats, we get the reverse transcriptase, we get the spike proteins for the outside of the capsid. All of that is produced from the host chromosome using that viral DNA that's been spliced in there. And so we can butt off these viruses. And again, the thing that's most nasty, we talked about this with the immune system, but the thing that's most nasty about HIV is that this cell that it is infecting is a helper T cell, whose main job is to detect infections, especially in your own cells. So this cell doesn't do that anymore. This cell makes viruses, and the viruses infect other T cells. And pretty soon, all of your helper T cells whose job it is to find viruses inside of cells, find cells with something going wrong inside of them, they are all doing this instead. HIV is called a retrovirus. And again, we call it retro. Retro just means backwards. And it's going, instead of DNA to RNA, it's going backwards in that central dogma from RNA to DNA. And that's the retro part. So COVID-19 is a new virus, right? This is not something that infected people before. And this happens all the time, right? Viruses come from somewhere and suddenly appear and start infecting us. And basically where they're coming from evolution. They're coming from other species, and they are getting mutations that change that little spike protein and allow them to bind to new receptors and therefore infect new species. So whenever we encounter other species, which we all are always doing, we are exposing ourselves to mutant versions of the viruses that these species carry. And we do the same thing to other organisms. We give our viruses to other organisms as well. And it's because of this high rate of mutation that's the fuel for evolution, right? It gives them the variation that makes it so they can end up with new traits. And the new traits are can infect humans. And that's a trait that, we're going to keep encountering. This is never going to go away. We're always going to have to be fighting against viruses. We're getting a lot better at it, though. Hopefully we'll learn enough from this one that when the next more deadly virus comes along, we can kick its butt early and keep it from, causing the problems that, say, the Spanish flu had, for example. Of course, the main source of variation in viruses is mutation, the original source of all variation. And if two viruses infect the same cell, this is really interesting because this is where we get very different viruses. You can take parts of the genome of two different viruses and make an entirely new virus. These are not uncommon. We end up with viruses that are hybrids of two different viruses from before that have new traits, infect new cells, are more virulent, or maybe survive longer on surfaces, all kinds of things. You can get deadly combinations of traits from combining viruses inside of cells. Most of you have probably been vaccinated for HPV. The reason for that is that HPV causes cancer, causes cervical cancer. And it's a virus. And it does what we've been talking about. This is a virus that inserts its DNA into the genome of a host cell, and it inserts it in places that are regulatory genes for the cell cycle. When it infects the cell, it disrupts the cell cycle and therefore can cause cancer. So this is why we vaccinate for it, because it's very easy to transmit. It's a virus that causes cancer very regularly. So I want to go through a quick summary of viruses, the main things you need to know. The general structure of a virus, you should know that. The protein coat around a nucleic acid, either DNA or RNA. So part of the protein coat of a virus has to bind to a receptor of a cell it's going to affect, right? This is chemistry. This is just molecules binding to molecules like we've talked about before. When it infects a cell, in general terms, it hijacks the cellular machinery to make copies of its nucleic acid and to make more of its protein coats. Lytic cycle versus lysogenic cycle. Lytic cycle, remember LYT, same word root as LYS. It means to split or to burst, right? It's going to make copies of itself and burst the cell open. The lysogenic, it's going to sit in the genome, right? It makes a copy of it and splices itself into the genome so that it can wait until conditions are right. Whatever viral trigger causes us to start becoming transcribed and translated so that we can make this virus again. And again, that's about the molecules that are in the cell. That's about transcription factors for the virus. Retrovirus is different because they're RNA and they're used to make DNA, which is a weird thing, right? They have this reverse transcriptase enzyme that allows them to make DNA from RNA. Cells have defenses against viruses, and oftentimes they are just enzymes that cut nucleic acids. Endonucleases, I think I've mentioned this before, endonucleases cut DNA or RNA somewhere in the middle of the strand. Exonucleases digest from the 5' end. We've talked about those when we were talking about the lifespan of the RNA. But endonucleases are used as tools when we want to cut DNA for technology. We want to splice together DNA or RNA, we use endonucleases. And those will come up more later. Just because it doesn't really fit anywhere else, I went ahead and threw a slide in here about prions. It's not something AP's ever asked about. Prions are basically a misfolded protein, and they're misfolded in a way that they corrupt the three-dimensional shape of other proteins of the same sequence. So you have a proper sequence of this protein, and then you have another gene that codes for one that folds a little bit weird, but it's the same sequence except a little bit maybe changed. And so you end up with this different folded version, and it changes the other ones into this different folded version. And so pretty soon, all of the proteins in that cell of that particular variety are all folded improperly and no longer functioning. And a lot of times they polymerize and they make these big, huge stacks of proteins that jam up the cellular machinery, make it so that cells can't do what they're supposed to do. And they act a lot like viruses, which is why I put them here. But you probably heard of mad cow disease, probably have not heard of Kuru or Creutzfeldt-Jakob. These are prion diseases. A lot of times they're found in the brain. They're proteins that are misfolded, and if you eat them or get them in your blood in some way, they will corrupt the proteins that are in these cells. So a lot of times these are in neurons, which is why you end up with, mental issues, cow madness and such. So that's viruses. Like I said, AP doesn't ask too much in the way of nitty-gritty details. We've got a couple more presentations on genetic technology. I'm going to go to school and do a lab. And then we'll have a review day, probably, and a test. I think we're looking at, I can't remember I said the date today, but it was right at the end of April. I think it's a Wednesday, the last Wednesday of April is when we're looking at having that test. It won't be huge, won't be that big of a deal, but we'll worry about it when we get closer. Hang in there, guys. You're doing great.